In recent years, genetic technologies like CRISPR, Cas9, mRNA platforms, and synthetic biology have entered mainstream medicine. While presented as therapeutic breakthroughs, many of these interventions now blur the line between healing and biological redesign. Historically, therapy aimed to restore physiological balance. Today, genetic interventions often reprogram immune functions, edit germ cells, and impact inheritable traits, pushing medicine toward modification rather than restoration. Economically, the human genome is being transformed into intellectual property. Companies now patent immune memory, protein behaviors, and even gene editing tools. This commodification shifts biology into market territory, raising serious ethical concerns. Some treatments rely on immune mimicry, introducing synthetic molecules that simulate disease to train the immune system. While effective short-term, they can induce chronic inflammation, immune fatigue, or autoimmune risks when repeated or poorly regulated. Beyond clinical use, gene editing has potential dual-use risks. Defense studies suggest genetic tools might be applied to alter fertility, neural behavior, or resistance to disease in populations, without visible weapons. These technologies could become instruments of silent biological control. Although tools like CRISPR are precise, off-target effects remain common, including unintended mutations, mosaicism, and disruptions in immune or neurological systems. In germline editing, such changes could pass to future generations irreversibly. A striking case was the 2018 announcement by Chinese researcher He Jiankui, who edited the CCR5 gene in twin embryos to resist HIV. While presented as protective, this unauthorized human experiment crossed ethical red lines, revealing how easily therapeutic intentions can shift into eugenic territory. Global bioethics councils, including the National Academies of Sciences, USA, UNESCO, and the Nuffield Council, have called for clear boundaries, somatic editing only, and a moratorium on germline modifications for non-therapeutic purposes. Philosophically, the debate centers on genomic dignity. Editing the human genome is not just a technical act, it reshapes identity, inheritance, and the natural trajectory of evolution. Treating the genome as an editable canvas risks erasing complexity, diversity, and resilience forged over millennia. There is urgent need for a global genetic ethics charter, ensuring a binding ban on germline enhancement, independent oversight of gene therapies, intergenerational consent principles, full transparency in clinical outcomes, legal protection for whistleblowers in the biotech field. The human genome is more than code. It is a historical record of adaptation and survival. Medicine must not become a gateway to invisible forms of control. Ethics must guide science, not follow it.